because of the fact that microscopes have gotten better, and really to understand in regenerative medicine specifically, the questions we get asked on a regular basis are, I've built 3D bioprinted, developed, matured a regenerative construct, a tissue of some type, and I've implanted it into a model animal, let's say a pig or a sheep, in which we're trying to understand a heart valve or an orthopedic application in which we've engineered a cartilage construct. The question is how does that tissue incorporate into the organism? That's the first question. How does the organism react to that construct? And what in fact is going on in that construct in relation to the organism, the animal system? We use an animal model here. We've started asking those questions, but we're incapable of addressing them, because what would happen is they would put this construct into an animal for 3 to 6 months, remove it, and give it to us, and we would be relegated to really taking that tissue and cutting it into individual slides and sections in which we've now removed the dimensionality to it, and then imaged it on our sophisticated microscopes, and then forced or guessing to try to reconstruct what an actual tissue looks like based on a bunch of two-dimensional information. There have been a number of attempts at taking whole tissues and putting them under microscopes, but that's been plagued by two basic problems. First is tissues by nature are composed of a whole host of different lipids and proteins and liquids and all nucleic acids, everything. And then when they're put into a structure, there's extracellular matrix that's involved. It could be bone, there could be blood vessel. All these components basically absorb light or refract light in ways that make it impossible to take a traditional lab-based microscope and see any depth whatsoever. There has been some basic methods for the past 5 or so years to try to figure out how to essentially remove those light scattering or light absorbing moieties. And there's been a lot of effort on trying to what's called homogenize light reflectance. What we're trying to do with the methodologies we created is to be able to see through tissue at depth. I want to be able to take a whole tissue out of an animal, and probably a person eventually, and be able to see through that whole thing using a microscope that can give us cell level information, but then when we're finished, we have three-dimensional spatial information. So the equivalent is, I want a microscope that can look at the 30,000 foot Google sky view and then be able to zoom in all the way down to Google Street View. And the only way you can do that is to optically clear the tissue. So in 2015, this field kind of took off. There was another investigator at Stanford who kind of led the way of at least the thought of being able to physically do this. And he essentially took a tissue and put it in detergent. And that got rid of all the lipids, the fats in the cells. And that allowed light to travel much further. And he was able to look through mouse brains. He's a neurobiologist, and he was able to look through mouse brains. But that was really an aha moment for most of us, that the key is to homogenizing light scatter and essentially refractive index. And you understand refractive index because if you have a glass of water and you put a pencil in it, you notice that the light bends differently and the pencil looks bent. That's what happens to the light when we shine light through a tissue. It bends every different direction and it never comes back to the microscope. So we need to make it straight. So we need to be able to normalize or homogenize that light. And so what we undertook was essentially coming up with a much broader, a much kinder and gentler chemistry, and a chemistry that would be able to penetrate not just mouse-sized tissues, but large animal tissues. In our case, again, looking at pigs, sheep, these type of animals, because that's where the regenerative constructs that we work with are put into. But we developed actually a technology of optical clearing that utilizes two methods, it's a hybrid methodology that takes the tissue straight out of the animal And we actually fix this tissue using some proprietary chemistry, but we fixed all the tissue in 3-dimensional space. So the tissue basically is fixed, and then we run it through a pressurized system with our own chemistry in it that's not detergent-based. It has some alcohols and other chemicals in it in addition. That's the hybrid part. It's not only just detergent, which is what the first methods used. Ours has detergents and surfactants. They're mild, and then we include alcohols, which do the rest of the clearing. And then within, depending on the size of the tissue, anywhere between 4 days and 2 weeks, we can optically clear, essentially turn a fully intact piece of tissue see-through. And if you saw the presentation that I gave, we are now looking at non-human primates. So a great example is we have the hand of a monkey with bone, cartilage, muscle, tendon, fat. The whole hand is totally intact. And we put this hand into our optical clearing methodology. And within 2 weeks, the hand becomes totally transparent. Now, the bone's still present. It's just optically transparent. The blood vessels, cartilage, tendon, fat, skin, all is still present. Nothing's missing. It's just the light now no longer bends differently because of all those light scattering moieties have been removed, and refractive index, or the ability to match the inside of the tissue with the outside world, the liquid, has been resolved, and so this tissue becomes see-through. So we call this method INCITE. It's actually spelled INCITE. We weren't particularly clever with the name. It actually stands for Index Matched Clear Imaging Technology. Not a particularly clever name, but we went with the INCITE because using this method allows us to take a standard laboratory confocal or multiphoton microscope and actually see tissue at depth in full 3-dimension. And because of that, we've been able to actually image tissues, full structures, fully intact with all their matrix, all the intact architecture and structural information, fully intact. But now we have the Google Sky View, which is the whole tissue. And then we can zoom in, because it is a microscope, all the way down to the individual cell and see where one cell sits in relation to the whole tissue. So why is this important? So actually, one of the more important things that's happening right now is, while we know a lot about biology and molecular biology and cancer biology, and these things are really, really taking off, one of the things we certainly don't understand, at least the importance of all the cells interacting together in this environment to form this architecture, this structure. The premise now is to try to develop cell atlases. So essentially to map every cell in the human body in position with its neighbors and the surrounding tissue in structure. And the only way that's going to happen is using these methods. Right now, the scientists are doing what they normally do, which is they take the tissue out, they grind it up, they do RNA extractions, DNA extractions, do RNA-seq, and the expression profiles, or the expression genomes of these tissues are well described. Cancer in specific is becoming well described by taking a tumor and literally doing RNA-seq on the whole tumor. But what you miss is all the spatial biology involved with how important it is to be a tumor cell or tumor cancer stem cell in the niche, in its little environment, protected by certain cells, fed nutrients by a blood vessel. All those things probably contribute to drug resistance and chemotherapy resistance, but because what you're doing is taking the tumor out in whole, grinding it up, and putting it through a machine, you have no idea where all this information is coming from. You just have the information. So the cell atlas concept is going to allow us to marry those two technologies together and bring the understanding of, oh, right, this cell has this gene expression profile. This is the drug-resistant cancer cell. This is what we need to go after. And so INCITE has allowed us, at least in the regenerative medicine space, to say, okay, we've put a 3D printed heart valve into a pig. At 3 months, is it normal? Does it look normal? has all the extracellular matrix aligned in the appropriate manner to withstand shear forces of blood, have the tissues formed around it to make sure the piece of 3D construct we printed is fully integrated into the heart. And these are the kind of questions that we can address now using this INCITE.